Oops. Just. Okay, just give me a second. I'm trying to figure out how to change my slide. That somehow seemed difficult. Okay. Are you able to see anything on my screen?
No, ma'am.
All right. Just give me a second.
Ma'am, you're on mute.
So is this okay? Are you able to see my slide? I know that you see the whole window. It's just that I'm not able to manipulate my slides if I go on the play mode for whatever reason. Is this okay? Are people comfortable with this?
Yes, ma'am.
Yeah, that's fine.
Yeah.
I don't know if any of the students have any quick solution for this. There's some tech savvy.
Okay, let me try once again.
Oh, yeah, this comes up.
Just give me a second.
So
All right.
only the transition is a problem, is it?
Yeah. All right. Let me see. I think it's going to work now. All right. Sorry about all this. I mean, just changing the system can sometimes throw you off. So I'm just giving you a little bit background of what I am and what I have done to be able to talk to you about the ideas that I'm going to be talking to you about. So I'm somebody who, for the most part of her life, has trained as a biologist and was on the way to be a blue-blooded scientist. But towards the end of my PhD, I sort of dropped my biped and I decided to pick up a paintbrush. And since then, I got the opportunity to work with different groups, to talk to multiple artists and sort of understand how science deserves stories and how that connects and resonates with different audiences. Like Kiran mentioned in my introduction, the most sort of highlight or biggest highlight or the turning point in my story was when I got to do this interesting postdoctoral fellowship at INSTEM, where my daily job required that I was painting in the lab instead of doing some or the other kind of experiment. And since then, I have worked on different kinds of sort of outputs for science communication and science art, whether it's children's books or zines. That's my favorite medium, and I'll talk briefly about it. And lately, I've also ventured into video making and like someone mentioned recently, self-published a book. And the other thing that I'm actively involved is that I train a lot of young scientists and students into with the ideas of science communication and science illustration in particular. And very recently, I've joined the visual communication department at Srishti Manipal Institute, where I'll be working with art and design students and hopefully bridging the gap between science and design. And my talk is going to be in 3 parts. I will talk about images in science, images in science communication, and images in science education. The third one, maybe we can skip if the time doesn't really allow us to do that. So I'll just begin by sort of thinking about science with all of you. Before I move on to talking about all of the visuals, I just want you to think, and maybe if you want to share, you can share as well. What are the ideas? What are the images that come to your mind when you think science? Like, what are the iconographies that you associate with it, or what are the images that you're so used to seeing in the work you do or in the education you're getting, that you associate with the idea of science and technology, and just spend some time, sort of, I mean, not now, but like later, maybe just spend some time reflecting on those. And science, and hence the images in science, are ultimately a construction of both the mind and the hand. And I mean, science is also a practice and a process which involves both of these faculties. And we as humans are able to perform this practice because we are curious by nature. And this curiosity has led to a lot of, well, for the want of the better word right now, scientific progress. And images have and visuals have always been a part of it. So let me start by sort of enumerating some examples from 16th century and that period of 16th to 19th century. So in that time frame, the easiest way of sharing science was the medium of the textbook. So the images you have on the screen here are what were part of textbooks. And like now, the way we disseminate these images, of course, the mediums have changed. So the first example is of looking at the highly detailed anatomy of the body as it is. And the second one is a hypothesis of how the planets might be arranged with sun in the center. So this is the one that talks about heliocentric view of the universe. And the third one is a documentation of what we now understand and are called as biological cells, as viewed through a magnifying lens or a microscope. And then we have also examples like these where Newton described relationships between geometries or relationship between different prisms and the resulting light behavior as a kind of schematic for us to appreciate what he was thinking about and looking at. And then there are also examples where you have experimental setups documented as what would be a photograph, as drawings of how the apparatus is arranged. And then this last example of Dalton is the most interesting one in that context, because it now is using a different language of symbols. It's defining a set of symbols to talk about different atoms and molecules. And these were all hand-done illustrations. I mean, in some sense, hand is always going to be part of all image production, but these were these actively involved practice of drawing in some sense. And then a little later in 19th century, the dimensions of how we made these images also changed. Now we had newer technology, photography, X-rays, and electron microscopes to capture data that would otherwise be inaccessible to the eye and hence to the hand. And so now we had different ways of recording them and documenting them. And oops. Okay, I'm back to my situation where I'm unable to move my slide. Okay, no. And then in the 19th century and 20th century, what changed was that we had 21st century rather, we had computing power, and now we could assimilate huge amounts of data and feed that to a system to generate a resulting image. So what you see on the left is hopefully an image that all of us have seen, which is that of a black hole that captured the imagination of scientists, students, and public all at one go. And similarly, the image on the right, which is one of the first few images which talks about the density of neurons and connections between them and their interlinking. So it's a cross section of the brain, and this was also an image that captured a lot of public attention. And so both of these images are both a progress in science and in science communication and science storytelling. So images through the early centuries, like since when science became a practice to up till now, like image creation and image analysis has been at the heart of scientific discovery. And whether it's observation, whether it's hypothesizing, it's analyzing, interpreting, or representing science, visuals have been a part and parcel of scientific practice, along with the very obvious words that we are all trained to write and read. Uh, so now I'm going to talk about drawing first for science. So in the previous section, I introduced the idea that images are used in the whole scientific endeavor, but whether they are done by hand or whether they are done by captured using technology, they're both part and parcel of science. But because I'm an illustrator, a lot of my work is thinking about drawing itself as an activity for science. And that's what I'm going to do talk about now. So one of my favorite example is this example from Darwin's notebook when he's thinking about how different life forms are related to each other. So he starts his sentence with, I think, and then ends not in words, but in this doodle, in this image. And this image is one of the most fundamental images. And when we try to think about the idea of tree of life and how things are related to each other, or how some things become extinct, and that there is a lot of diversity. So like I already argued for, visuals are intricately involved in scientific communication and in the very construction of scientific fact. Now, observation is the fundamental base of all scientific research. So this image is part of a larger body of work by this, I would call scientist and illustrator, Maria Sibylla Merian, who spent a lot of time in the forests of Suriname and produced this huge body of work where she represented plant and insect relationships. And what was interesting about her process and her work was that it also sort of clarified the idea of how caterpillars and insects are related to each other via the life cycle, because there were previously arguments that indicated that caterpillar was a different species and the moth or the butterfly was a different species. So her work was amongst the ones that sort of brought it all together. So she spent a lot of time observing and documenting on small pieces of paper. When I say documenting here, I mean by drawing and additional annotated notes, drawings of different life stages. So she would follow the plant as well as the insect from egg to larva and so on. And then at the end, compile all of these images into a single image where all the life forms and all these stages of, like, say, blooming or fruiting of the plant were put together in one image. So she took the creative liberty there of sort of packing a lot of time together in one image, but she emphasized on the idea of form and relationships. And all of these details in terms of how they look and what they color, she tried to be as true as possible to what the reality was and what she saw. So seeing is an art which in some sense should be learned. And now this is the work of Robert Hooke, who was amongst one of the first few people who used the microscope to look at biological specimens and a lot of other specimens, but then most importantly, biological specimens. So in his book, Micrographia, which was the result of all the work he did with the microscope, he, in the preface, he justifies his actions and says that what he did essentially was just being a faithful assistant to microscope, which is really the instrument that did the work. And he said that to be able to do the kind of work he did, All he needed was a faithfulness to the microscope by the way of his eyes, mind, and hand. So all these three faculties had to be honest to what he was seeing and what he was drawing. And in his process, also, what he did was he would capture multiple specimens and look at them from different lighting angles, different magnification powers, and so on to finally make this one composite image, which indicated what the specimen was. In some cases, he did make multiple drawings of the specimen as well, or showed us multiple specimens of the same kind. And what it says is that vision is not just a mechanical recording of elements, but rather an accumulation of significant structural patterns, which is kind of something that we are also looking to when we're practicing science, which is looking for patterns and then try to build hypotheses from there. And now I will talk about a little bit of my own work. And I just want to say this explicitly, that you might feel a little overwhelmed because this is all like a molecular structure thing and maybe you're not familiar with it, but trust me that if you keep an open mind, I will take you through it in a way that you will be able to at least relate to the point that I'm trying to make. Right. So Renaud, a collaborator of mine on this project, has once said that drawing enables making of images that are more abstract than a photograph, but have more depth, especially with the relationship of space and time, and are far more detailed and accurate than the schematics. So don't worry about the name. The name is called zine. And what's the role of this zine? So you see these pigmented cells on a fish scale here. And you can see that like this spreads and then comes back this pigment within the cell. So this particular character that we're talking about, dynin, is the one that brings it, like shrinks the pigment to the center of the cell. And I was interested in, and rather we were interested in understanding this molecule, and I chose drawing as a medium. And this was done in my postdoctoral lab, where this molecule is a sort of subject of interest for the molecular chemists and biologists that work there. And we don't have to sort of go into the technical ideas about what this motor domains are and all of that. But the idea was that we had the technical data, the crystal structures taken by multiple people. And I started to use that data to understand the form of this molecule, how it looks in its volume. I even made some clay models, and I did some sort of reference-based drawings, some memory-based drawings, some refined sketches, some loose sketches, and then started to understand that how this molecule really works in space. And then I had another challenge here, which is that this particular molecule that I'm talking about, it does not work singly. It works in a pair. So the two molecules of same kind work, and they walk along this particular track in the cell. And it doesn't matter what these tracks are called and all that. And so once I understood how it looks like, I sort of started wondering about, like, when they walk, how do they look like? Do they look like us? Do they look like caterpillar? What's the situation? And so what I started to do was read up all of the literature, including the visual and the non-visual, to figure out their behavior and their relationship and patterns from different kinds of data, whether it was the structural data, or it was biochemical data, or it was cell biological data. And I sort of made these drawings where I tried to put them together. And as I put them together, I realized that, certain things I wasn't sure about how to place them, or what was the angle I should be drawing them in. And that meant that I had to look for more information. And so this became kind of an iterative process where I was looking, drawing and asking questions, and then So trying to find more information about this. And then what happened towards the end was that I was able to make these drawings, but then these drawings also had a lot of uncertainty with them. So I had a summary, but I also had a lot of questions. And these questions are, in some sense, also new to the field. So by the way of summarizing a body of work through the medium of drawing, I was able to ask new questions, which is really like the starting point for a scientific inquiry. And then this didn't really stop there. Once I understood how they looked like when they were together, I then also decided to figure out how they might work. And from the scientific data, I knew that there was some literature which talked about that they walked in small steps, and some which said that they walk in longer steps. And what I'm going to do is just try to play, if the technology works with me, is play, multiple slides quickly. And what you will start to appreciate there is, so I started to understand like what those permutations and combinations might be on a graph paper. And then I just basically drew them. And so what you might see from all these different kinds of walks that I tried to put together here is that different walks meant that in the same number of steps, there were different distances covered. And that meant that if we have the speed data, then we can start to figure out that what is the more probable model to put in respect of how this molecule works amongst all the possibilities that are there in the literature. So in this case, we were able to predict something as opposed to asking, right? So this is one of those case studies where drawing was used in a way to generate scientific inquiry and maybe propose models which were more suitable to explaining the existing body of data. I didn't say this earlier, but if any one of you feels like asking a question at any point of time, please feel free to do that. And if any one of you feels like you would like more clarity on something that I've been discussing, happy to sort of converse about that as well. So what's really cool about drawing is that it's free from the pragmatic constraints of the experimentation. When you think about your experiments or your hypothesis, just because you understand the methods of your field of study, you will be limited by that. But if you start thinking about an activity like drawing or mind mapping, there's no limit to it because you are only limited by your mind there. So it allows you to sort of unlock a certain sense of creativity and unlock a certain potential, which is otherwise not available to you. Right. So now I'm going to switch gears and then talk about drawing for storytelling. And you can also think of this segment as drawing for science communication. And the first example that I would like to bring is of Irving Guys. He was an illustrator who drew a lot of protein structures. And in fact, the first protein structure we had, which was for a protein called myoglobin, doesn't really matter. And this was, his drawing was there on the cover of, I believe, Scientific American. And then so I have two images here, one that was drawn by Bill Gates and one that's of the same family of molecule taken from this database that we have for protein structures called Protein Data Bank or something. I don't know. PDB is how we call it. Right. So the way and you will see that there are certain differences. I mean, if you just pay attention to the form, you will see that there's like a subtle difference in how the molecule is represented. And that comes from Irving's process. So what he did was he actually built physical models of this protein, of whatever the proteins that he worked with. And then he photographed from multiple angles and used those references to draw. But in the end, he said something very interesting. He said he wanted not to draw the protein exactly as it was, but to show how it worked, which means he relied on this process that he called selective lying, where he distorted certain angles, certain bonds, and exaggerated some parts to show what's important in the molecule that's relevant for the audience to sort of think about. And he said that this is one of the most important trait of a knowledgeable artist. A computer can draw a protein like the one on the right, and given the right set of coordinates, but who would tell it what to show? So here he emphasizes on the role of an artist, an intelligent artist who understands the science in being able to communicate what needs to be communicated. And I think what he's trying to say is essentially the idea of storytelling, where you understand your audience and then you sort of decide, you make choices for them to enable clarity and weigh in on clarity more than accuracy. Right. So, okay, oops, sorry, that went quickly. So now I'm going to share my sort of tries with science storytelling. So we recently came out with this book called Actually Colors Speak, which was also another postdoctoral project that I did at INSTEM with my PI, Sirajuddin. And the idea for us was to make science accessible. So we kind of did a Google-based survey of what are the popular science books out there. And most of the books that we saw were related to the ideas of astronomy or ecology. And we were our cell and molecular biologists, and we felt that that's a niche that's not being worked on enough. And particularly from India, we didn't find many really cell biology popular science books. And so we decided to fill the gap and make one ourselves. And we chose the idea of color in animals. And I mean, that was really just a decision that was based on the understanding that this is because we were going to make an illustrated book, it's a theme that resonates well with the audience and will also allow us for the possibility of making some cool illustrations. And so during this process is when I also had to figure out my pattern of selective lying. So for this entire book, it's heavily based on scientific data, available images, electronic microscopy and other microscopies and other biochemical data. So again, in this process, what I had to do was read up a lot of literature, look up images that were already available, and then transmute them in some sense to a format that I could use consistently throughout the book. And then figure out what are the features that I want to highlight and what are the features that I can maybe keep on the page, but subdue them either by making them, sort of drawing them with paler colors, or by increasing their transparency, or by even playing with scale. If I needed to show a certain cell type or more sort of prominently, I might play with its scales or its numbers and repeat it on the page enough number of times so that your eye definitely pays attention to it. And the other thing that this is to do with images, but the other thing that we did in the book was that we decided to tell the story from the point of view of the instruments that one would use in a lab that probably works on color. So we have a binocular, a camera, and a microscope, all talking to each other from their point of views. We understand what a camera can do. We understand what a binocular can do. We understand what a microscope can do. And we use we give them an agency of sorts and have them converse with each other and tell us the story of different animals and their behaviors and how it is all dependent on cellular and molecular processes. So we go from outside the animal to inside the animal, and then further inside the cell. Sorry, that was a wrong arrangement of the slides. But and some of the other ideas that we worked with in the book was that we tried to explain the phenomena. So though we were biologists, there's actually a lot of concepts from chemistry and physics that we had to incorporate in the book, so as to make the concept clear. So placing, and the need of our art audience before before content, like our expertise and our content. And the other thing that we were trying to achieve was to navigate across scale. So like I mentioned, from outside the scale of the animal to inside the cell. And that was kind of like a distance of not the distance, but like the degree of difference is kind of like between Mount Everest and the coin. So that's the scale we covered. And what the other cool thing that we've done in the book is we've sprinkled these QR codes here and there, which allow our audience to kind of go on and read more if they would like to. So yeah, I would recommend that you look up this book if you're interested, and happy to also talk about it more later if there's a space for conversation. Now, switching my gears again, I'm going to talk about the idea of drawing for learning. And in this context, I'd like to bring up something that a scientist, Waddington, said, that science is something more than a collection of conceptual or practical results. It's also an activity, and its practice involves, as a very important part, the exercise of faculties of the insightful perception of natural phenomena. So this is a phrase that's sort of hard to unpack, but also very interesting. Insightful perception of the natural phenomena and of imaginative creation of new process, which is kind of similar to what I was trying to say when I was talking to you about that molecular motor that works, is that when we were drawing, we were able to come up with new ideas that wouldn't have otherwise not been experimentally possible. So this is an activity that I used to do often, where I would ask people to draw based on prompts. So this was with a set of biologists in training, people who were doing their BSc in one or the other kind of life sciences. And then I just asked them to draw a cell. So these are students who have had access to sort of rich textbooks and so on, and are currently in the process of learning more about the cell. And then on the right, I had a set of images where I asked non-scientists, non-biologists, people who last studied biology, a decade ago, when until their 10th standard and so on, to also remember what they, thought of cells. And some interesting observations came about, like how cell was still circular for most of people. And those who were pursuing biology were able to remember more details about it, whereas other people just made it a circle within a circle with some speckles around it. And the details were not necessarily something that they remembered, except for nucleus and mitochondria in some cases. And all of these drawings actually were conveying the idea of a flat cell, of a 2D cell, in most cases. And so I wanted to break that perception. So I worked on a series of zines. Zines is another media that I work with a lot apart from books. Zines are, to my mind, essentially free from editorial constraints, mini booklets that you are free to make yourself and distribute as you would like. So I made a series of zines about the idea of what a cell is. And in one of the first one, because I was coming from that context, the first idea that I wanted to communicate was that a cell is actually a 3D object. And to sort of I came up with this visual metaphor. Now, orange, the fruit we eat, is something that we look at from the outside, and we look at it on the inside, and we slice it up in multiple ways, and we understand how it might look like. So that's a form that we understand. So what I did was I placed an orange and I placed the cell side by side, and then I sliced through the orange and I sliced through the cell, and then I showed how the orange would look, and then I showed how the cell would look. And at the end of the day, at the end of the zine, I also ask people to sort of draw and imagine what their cell phones or some other common object would look like from the inside if they were to sort of slice it up from different point of views. And what was interesting about this zine was that I was able to, I had the good opportunity to sort of show it in scientific conferences as well as show it in public spaces like the Comics Fest. And so which means I had access to different audience. I had access to scientific audience as well as I had access to a non-specialist audience. And interestingly, a lot of relationships that these audiences built were really the same, except the ones who were trained in sciences had the vocabulary to name it. They could call mitochondria, cilia, cilia, but the untrained audience didn't have that language. But just because the visuals were based on scientific data and were fairly accurate, they could see the relationship between like how this particular form, like the mitochondria is always close to the boundary of the cell and so on. And there were like unexpected metaphors as well. So in this slide, you see the seeds on the orange, and then you see the nucleus, which is where the hereditary material is sort of packaged. So this, like there were a couple of people who brought up this unexpected metaphor of heredity, sort of playing together both in the orange as well as the cell. So by presenting this zine, this new way of thinking and this new way of looking, I was able to break that sort of misconception about cell being a 2D sunny side up like of object, like something like a sunny side up, and build dimensions to it and call it a 3D object. And apart from that, there were people who took away more than I had sort of imagined conveying. And then we have also, especially in collaboration with people at CCMB, we've also used zine as directly as a teaching material, where we developed a set of zines to educate young students about different ideas. This one is one amongst my favorite, which is called Stuff of Life, where we talk about that how most of the molecules that are within our body or most of our life forms are just these few four elements, the carbon, the hydrogen, the oxygen, and nitrogen, and how just these four elements can make these complicated molecules and these complicated bodies that are living. And so like there is a transition happening somewhere from elements to living elements, and that's something that we talk about in this scene. So we were able to generate a sense of curiosity and wonder in the student body that looked at this work. So here again, we used images as the sort of fulcrum to convey these ideas. And I mean, though it's all scientific, they don't look scientific, and that was kind of our idea in this sort of teaching tool, right? And like I mentioned, that I do take a lot of sort of workshops and courses with students, and this here I used drawing as a new way of looking. And what we did was we basically collected a lot of plant material, flowers and leaves and whatnot, and we drew them. And we drew them first by just looking at them, how we saw and thought their shape and form was. So the literal object was in their hand while they drew this. And then the next activity, what we did was we measured, and then we drew based on those measurements. And then we looked at what's the difference between what we perceive and what the reality is. And so here we used drawing activity as a tool to break existing perceptions. And this is with something very familiar object. We're not looking through the microscope. We're not like looking at some unexpected material. We're working with the plant material, which is something that we observe on a very regular basis, or hopefully so. So it's part of our like inbuilt perception of what our world is. And by just having drawing activities, we were able to change that setting of what we think we know and what we know and what is, right? And in this in this activity, what we did was we showed some biology students an image that they are most likely to have seen, which is at the bottom corner of the slide, which is a 2D cross section of a rice plant stem. And it doesn't matter what the material is really. And then what I sort of tricked them into doing is that once they had that very familiar to them, 2D image, I asked them to imagine what it looks like in 3D. Like imagine you are drawing a chunk instead of drawing a thin slice. And then if you look at these images, you will see that how different students imagined different versions of what this thing actually looks like in 3D. Which now in that activity, what we ended up doing was sort of figuring out how one projects from 2D to 3D. But then you could also think about this also as an hypothesis building activity, not necessarily in this context, but in any other context. When you change the form from one setting to another setting, you allow yourself to ask sort of a lot of new questions that you would otherwise not be able to ask. Yeah. So in conclusion, drawing allows you to do a lot of things, and visuals have been part of scientific endeavor for a long time and continue to be so. And illustrators are cool people because they're trying to ask, observe, hypothesize, predict, infer, communicate, evaluate, and do science really. And with this, I would like to thank you for your attention. Thank the people of Minaj Lab and Minaj and some other collaborators with whom we did the protein drawing work, several collaborators and people who have done an amazing body of work before I got to doing it. Yeah. Thank you. Happy to take questions and comments, if any.
Thank you, Dr. Vipsa. It was amazing journey. So the floor is open for questions. Students or faculty, you can raise your hand and maybe Kiran can moderate.
Yes.
No questions. Okay.
There is a question from Shashwat Gai. You can ask.
Hello, good evening, ma'am.
Hi.
So I am currently just started my journey into material ecology, which basically talks about the dimensional mismatch that we have between the environmental space and the conceived design space. So which basically talks about how we should look into ecological mismatch as well because of the dimensional mismatch that happens in design. So so I was just wondering, have you looked into any FIM models or similar kind of models into dimensional which talks about dimensional mismatch in your work?
No, I can't say that I have, but I would be really curious if you were to give it a shot, or maybe we can figure out ways of trying something together. Yeah.
Yes, I would love to speak to you probably separately. I'll write you.
Yeah.
Ma'am, I would like to ask a question. This is Kiran.
Yeah.
So you mentioned in one of the slides about the orange and its cell on the similar slide, and how I'm exactly not sure how is it helping out or helping our imagination skills or improving. What kind of improvement are we like having in representing that?
Yeah. So the biggest fundamental idea that I had at that time was, then this is based on my own activities with science and non-science people, and also some interactions with teachers, where the perception of the cell is like, it looks like a sunny side up, where it's like a circle within a circle, but it's like a flattened object. And people don't view it as 3D object. So that metaphor of orange, which is, and that happens because the diagrams that we have in the textbook show cross-sectional drawing, which is kind of like the orange cross-section, the slice that you saw. And so my idea was to build a sort of relationship with something that we are familiar with and something that we're not familiar with, which is the cell in this case, both 3D objects, which we are used to looking at its cross-sectional form. Yeah. I think
Okay.
Pankaj had his hands up.
Yes, ma'am. So you can ask the question. Maybe he doesn't have a good internet connection.
Yeah, sure.
Uh,
So I'm just on the chat sharing some prompts that you can Google. So on Notion Press, you'll find the book that I was discussing, and maybe it's something that will interest you. And then if you just Google CCMB zines, you will see a lot of zines. And also on my website, there are a lot of zines and the book. So feel free to look them up if it's something that interests you. Yeah.
if students have doubts, you can also put them in the chat. We'll read them out. Yes, Ram Prasad.
Thank you, ma'am. It was a very interesting presentation. So actually, ma'am, I just wanted to ask, what are certain things which you have found it very difficult to draw or communicate through images? Like it is something very difficult, or
Right.
it's yeah. I just wanted to know about that.
Yeah. Thanks for this question. So one of the things that I still don't have an answer for, really, but one of the things that I have particularly found very difficult to indicate is time. Like we talk about life cycles or cell cycle or anything like that, like a process where you're changing from state 1 to state 2 and state 3 and so forth. And while it's very easy to indicate what those state 1, state 2, state 3, and state 4 are, um, the time relationship is often an arrow. And that arrow, I feel, is a difficult thing to comprehend. And also what happens is that one assumes that the time transition between state 1 and state 2 and state 2 and state 3 are equal because the arrow most likely you will draw are equal, which is not necessarily the case. Sometimes you will write the data that this takes place in few hours and this takes place in few days. But when you communicate science or you're learning science, that's not a data that you instinctively remember. So if you ask me today as a trained biologist as to what's the time gap between, let's say, the G phase of cell cycle or S phase of cell cycle. I mean, you don't need to know these phases, but I mean, just this, the state 1 and state 2 that I understand very clearly, or at least so I think, I can't answer. And So time is something that I've always found very challenging to sort of comprehend and hence also visually indicate. And I think if we were able to find a visual cue that allowed us to think about show time clearly, then comprehension of time will also become easier. And the other thing that I would say is that like I've been a biologist. I mean, I'm a biologist. So the things that I'm looking at are real objects in the sense that there is a touch feel to them. And very recently, I've been thinking about some ideas from physics and chemistry. And in fact, my next work hopefully will be about atoms. So now I'm thinking about how to visualize atoms. Now, for atoms, there are multiple different kinds of visuals, and in atoms is something that we're not literally seeing or not literally seeing the inside of the atom. And the ways we can understand them are very different from the way we can, let's say, look at a bacteria or things inside a bacteria or a protein. So how do I visualize something that I don't understand in the same framework that as I understand biological specimens? So that's something that's also interesting to me, and I'm hoping that maybe in 2 years or 3 years I'll have an answer for you about that. Yeah,
Thank you, ma'am.
thank you.
There's a question from Shivali in the chat. Do you think that this profession of science illustration is something you can make a good living out of? And have you met any students who aspire to be one?
Answer the first question, it's going to be a tough road. I mean, I always tell, and the second, yes, very much yes. I train a lot of students. I've worked with a lot of students. I do a lot of workshops and training exercises, and some of them are, sort of on their way to becoming science illustrators now. And there are a lot more that I haven't personally worked with, but I know are on their way to become science illustrators. So a lot of young students are aspiring to be one. Making a good living out of it will eventually be possible, but the road to it will definitely be of struggle. So if you have the privilege of being cushioned in some sense financially, then yes, it's worth thinking about it. Yeah.
Okay, ma'am. Thank you.
Welcome.
Ram Prasad, you can ask your question.
Ma'am, you gave an example wherein you studied various I don't know whether it is proteins or molecules, and then you came up with lot of drawings, and then you got questions based on your drawings that okay, could based on the patterns you observed in the drawings, could this be like in the real situation also, like you got certain questions.
Yes.
So based on history, if we observe, are there any instances in history, scientific history, wherein some scientific discoveries have happened uh, because when the scientist or when someone was when an artist was putting what has happened into an art form, they got some clues, and based on that it helped in some discoveries. Has it happened any time before?
Yes, yes, many, many, many times. I mean, the first obvious example that comes to one's mind is Leonardo da Vinci. Often, I mean, of course, it's very hard to differentiate between Leonardo the artist and Leonardo the scientist because he embodied both of them, those practices. But I'll give you another example, and I forget the name of the scientist and the theory, but this whole idea that all the continents on our planet, they came from a single landmass, and so that happened because of, again, like an image activity where the scientist in question sort of figured out that, like, if I were to chop off all or draw all the shapes of these continents and then put them together, they kind of like start to look together like a landmass. So in his case, actually, that argument came before his hypothesis. So because he made that, playful observation, he came up with a hypothesis. So the act of visualization happened prior to the hypothesis, if that makes any sense. And even in Newton's case, there are a few examples where he theorized about the behavior of light in certain experimental setup, like wondered about the possibility before actually doing the experiment. So there are these Newton's notes that are available online, which indicate some of this pattern. So a lot of scientists, even Darwin's example for case, for example, in his notebook, which is like much before his work, Origin of Species, he wrote that I think and then drew that tree. And so like thinking and drawing for a lot of people have gone hand in hand. And so thinking has led to the hypothesis or the experiment that they would work with eventually. Yeah.
Thank you. Thank you, ma'am.
Thank you.
There's a question in the chat from Anand. He's asking, in this era, data is ubiquitous in all walks of science, especially in social sciences. Do you consider data visualization as an art? If so, how do you compare it with drawings and the attributes you mentioned in your lecture?
I would actually place drawing and data visualization in parallel, and they all form like they are all part of the bigger umbrella of the visuals and science. And I would say that in a lot of cases, for example, the same holds true that once you have a lot of data, you are able to ask new questions, or you are able to identify new hypotheses. And so I think the same thing is possible with both. And when it comes to data visualization, I would actually like to point out that like all of us, including myself, we rely on graphs and so on, like digital softwares to process it and analyze it. But there are artists even in the contemporary world and have been in not so contemporary world who have done this analysis by hand and have come up with drawings. The one artist that I would really like to mention is Georgia Lupi. I'm a big fan of her work, who works on a lot of data visualization, both of qualitative and quantitative data. And I would recommend that you look up her work, and since if you're interested in data visualization, as to how you can humanize data. So a lot of her work is done by manually interpreting the data in very specific Excel sheets and then drawing based on it. And when I say drawing, I don't mean it in the sense of the representational drawings that I did, but like coded drawing as one would do in sort of any kind of data analysis. Let me write the name if that helps you. Sure. I could be wrong with the spelling of the name, but I think if you Google it, it will show up. Yeah, thanks, Shashwat.
Okay, last call for questions. We have approached the usual time of 1 hour. So if there are any quick questions, please speak up. Otherwise, we'd like to wind up.
Yeah, and you can always find me on social media. On Instagram, I'm fairly active, and I'm called Self-Windows. So, yeah.
Okay, so if there are no more questions, let's give a big thanks to Dr. Iksa Jain for finding time and giving this new perspective to look at science and arts both. I believe, and traditionally we believe that the brain has two parts and each of them work on these, one for science and one for arts, but probably
No,
there
any
is this.
neuroscientist will tell you that's all incorrect theory, and
Okay,
it has
yeah,
no
I stand
scientific
corrected, but we have this unique example where she has mastered both. So very nice to share that perspective with our students. And I think the lots of questions that student came up shows that there was interest in this new topic as well. So thank you so much for being
having
here
me.
with us, although virtually, and big thanks from all of us.
Thank you. Thank you. Good evening. Bye, everyone.
Thank you.
Thank you.